I'm now going to introduce very briefly Roger Meacock, who I'm sure again is well known to many of you, a vet of many, many years' experience, but specifically and importantly a holistic vet, or perhaps it should be an holistic vet, specialising in horses, but also humans. And I think it's very important to consider the links between humans and animals. I know it's very easy and obvious to be perhaps a bit glib about that, but it is very, very significant. And I think Roger, in particular, has enormous amounts of insights into the direction of travel by means of examining what it is that's being done to animals in the name of, say, science or progress, and then seeing how that has an impact on humans. So please welcome Roger Meacock.
Thanks, Charles. And I'd also like to say well done to Sandy. If you want to see what she did in more detail, we did actually a series on UK column, which is titled Dear Farmers, and it's a series of 3 different programmes. So if you want to see more detail of what Sandy's gone into, then you can see it on UK column. So that's me, Roger Meacock. Natural Healing Solutions has been my name of my practice since 2000. So today I'm going to talk about PCR because it's important, so the modRNA potential for veterinary use, Bovaer, and a little bit on climate as well. So there's a lot to get through. It could do hours on each of these, so we're going to rush through as quickly as possible. So what are PCR tests? PCR stands for polymerase chain reaction, a test developed by Kary Mullis for which he received a Nobel Prize. It was developed as a clean laboratory test to detect a known sequence amongst other known sequences of RNA or DNA following gene therapy manipulation, or to detect a known mutation. And I've put the bold in because it's really important. There's a subtle, really important aspect to this definition, which is that it is detecting known sequences amongst other known sequences. That's really crucial. And I didn't get that straight away. So it identifies a selective sequence, which may only be present in otherwise unmeasurable small amounts, and doubles the amount present at each cycle of the PCR test. So the number of cycles used in generating a PCR result is important to know. So different primers are chosen according to the sequence of RNA DNA that is to be detected and amplified. So this is a little video that tells you how it is.
PCR is a powerful technique that allows us to make billions of copies of specific pieces of DNA. The first step in PCR is to open up the 2 strands of DNA to expose the genetic information encoded by the nitrogen bases, A, C, T, and G. In the next step, custom-made DNA molecules help us find the beginning and end of the DNA piece of interest among the billions of letters in the genome. These genetic bookmarks are called primers. The third and final step is to fill in the genetic information between the primers. An enzyme called DNA polymerase adds the building blocks of DNA called nucleotides to create an identical DNA strand. DNA polymerase works in our own cells to replicate our own DNA each time a cell divides. The first step in PCR is known as denaturation and is enabled by high heat. The second step is called annealing, and different primers have different and very specific annealing temperatures, typically between 50 and 60 degrees Celsius. The third step is called extension. DNA polymerase used in PCR works best at 72 degrees Celsius. In these 3 steps, we've taken our original DNA molecule and obtained an identical copy of a very specific fragment. If we repeat these 3 steps one more time, we'll go from 2 copies to 4. Repeat it again and we'll obtain 8 copies. And again, and we'll get 16, and 32, and 64, 128, well, you get the idea. DNA amplification by PCR is an exponential process. After 30 copy cycles, we will have 2 to the 30 or 1 billion identical DNA copies.
There we go. So that's PCR. We can pass an exam on it now. So the next slide is Carrie Mullis, who's the guy who invented PCR, and he's saying what we can and what we can't do with it. In the video, he's talking a little bit about it with regard to HIV, because that was when he was alive, when he was talking about it. But obviously, it corresponds to whether it's bird flu or any other pathogen that they might be checking for.
Misuse PCR to estimate all these supposed free viral RNAs that may or may not be there.
I think misuse PCR is not quite. I don't think you can misuse PCR. No, the results, the interpretation of it. See, if you can say, if they could find this virus in you at all, and with PCR, if you do it well, you can find almost anything in anybody. It starts making you believe in the sort of Buddhist notion that everything is contained in everything else, right? I mean, because if you can amplify one single molecule up to something that you can really measure, which PCR can do, then there's just very few molecules that you don't have at least one single one of them in your body, okay? So that could be thought of as a misuse of it, just to claim that it's meaningful. It is. No, there's very little of what they call HIV, and what's been brought out here by Philpott and Isai already, the measurement for it is not exact at all. It's not as good as our measurement for things like apples. An apple is an apple. You know, you can get something that's kind of like, if you got enough things that look kind of like an apple and you stick them all together, you might think it's an apple. But and HIV is like that. Those tests are all based on things that are invisible, and the results are inferred in a sense. PCR is separate from that. It's just a process that's used to make a whole lot of something out of something. That's what
Also,
it is. But it's not. It doesn't tell you that you're sick, and it doesn't tell you that the thing you ended up with really was going to hurt you or anything.
There we go. That's the key line, isn't it? It doesn't tell you that you're sick or that what you've got is going to be infectious. So what is PCR testing for? So as the video explains, only RNA and DNA with the correct sequence of nucleotides, which are just A, C, G, and T, that's all, will bind the chosen primer. So you get short primers, which are 18 to 20 nucleotides long, which provide faster binding but may have lower specificity, so they won't more things will cause a positive reaction, leading to false positives. Or you can have longer primers, where you've got 25 to 30 nucleotides, but they offer higher specificity, but you can get then folding of the primer or the DNA section that it's trying to check, which can obviously cause misreadings as well. So then you get false results from that as well. So if there's been a mutation within the primer sequence, then it won't be recognized. Or conversely, a mutation within a sequence that has actually then created what you're then looking for will also then be detected when it otherwise wouldn't be. And when we're talking about things like bird flu, which are frequently mutating, it sort of makes the PCR all that less reliable. So, okay, which of the following viruses are HPA, which is highly pathogenic AI, avian influenza, H5N1? Has anybody got any views?
No. They all look like man here.
The man here got it right. Answer: any, all, or none of them. If the section of the sequence of those recognised by PCR, and this is really crucial, they will, if they come up with a positive, they will say that's what it is. But it could be any one of anything else. But because it's come back as a positive on the PCR, it will be identified as what they're looking for. So what does a PCR positive actually mean? It means that the fragment of DNA that they've been looking for has been found that matches the primer. That's it. PCR cannot tell if the original RNA DNA came from a viable virus or how many of them there were. It cannot tell if the virus was capable of infecting the host and dividing. It cannot tell if there was a pathogen or a contaminant or part of the microbiome, or if it was mutated. It cannot tell if the immune system had already dealt with it and you just picked up a fragment of what the immune system had already dealt with. Without knowing the CT number, so the number of cycles that that doubling process does, it is impossible to know if the original sample was capable of being infectious. For COVID, it was above 24 cycles was considered that the initial sample was therefore incapable of causing infection. But for these tests that they're doing, they're frequently cycling over 30 and even over 40 cycles. So once you've got to 30, you've got 1 billion times more than when you restarted with. And if that's doubling each time, I mean, you can work out how many times you've got by the time you've done it 40 times. It's an inconceivable number. So an analogy of this is if I was to take a swab from my sofa and look for my dog, there might only be a hair there, but they're telling you when you get back a positive, not only is the hair there, but the dog is too. That is the system where we've got to now with what's happening with PCR. So what is the risk of a false positive? Where the disease prevalence is low, then the chance of a positive result being false positive is very high. And I don't just mean like 50, 60%, it's 90-something% chance that it's a false positive. This is especially true when looking for HPAI, high pathogen avian influenza, in species other than birds and or in the environment, which then provides false epidemiological results as well. So where there's a lockdown, an exclusion zone or a cull policy is in place, a false positive, as you know, has serious implications. If there is another cause of similar symptoms that can be affecting a population, then a false positive PCR means they won't go on to look for what has actually caused it. So it could be spraying, it could be vitamin E deficiency, which causes the similar symptoms, or you could even get some other pathogen as well. And it can, in an enclosed flock contracting HPAI, if there's no contact with the outside world, how has it happened? Common sense has got to come in here somewhere. If the police were looking at a crime, if there's no way for a criminal to get through the door, even if they can find a slither of that person's DNA in the room, and there was no way that they were ever going to be there because they've got an alibi as well, then they can't be convicted and they wouldn't get a result if they tried to go to court on that person. But that's not what's happening with the PCR test in the farming situation. They're not looking at the common senses. Even if we got a false positive here, how did the pathogen get in the first place? We've got to have some common sense. So how do we safeguard against false positives? False result figures should be established for all and any test in use, and they don't know them. PCR should not be used in isolation in a clinical situation. There was going back to that picture of the viruses, and I said which ones are which. A good example of this that actually occurred was the president of one of the African nations. When COVID was being looked for, he sent off samples from goat, papaya, various other things, and they all came back positive. So that tells you how PCR is being interpreted. As Carrie Mann says, it's not that it's being used, it's being misinterpreted to insinuate that there was an infection there which couldn't possibly have been there. And this is what we've got to worry about and warn against for use on the farms. So PCR should not be used in isolation in a clinical situation from a swab with a highly mixed microbiome source. For those who haven't heard much about the microbiome, it's all the bacteria, all the viruses, all the fungi and everything, all the microbes that live on us. And roughly 10% of me standing here is human cells. 90% is microbiome. So when you're taking a swab of that microbiome, which has got sequences which they'll have no idea what's present in those other microbes, the chances of getting a PCR positive, they've got no idea whether it came from what they're looking for or anything else. And there's a high probability it came from some other source. And so this is where we've got this problem. So the CT number must be less than 23 as well, and certainly not the 30 to 40 as is commonly used. It should only be used where there are clinical symptoms. You can't go in where there's no clinical symptoms and screen for it, because it doesn't work. They did something similar in the States where they're looking at cattle, and you know, they found it in an eye swab. Well, you know, if put it blunt, if a bird crapped in the feed trough and a bit of dust got in the cow's eyes, it put its head in the trough to eat. Of course, there's going to be something positive, you know, possibly detectable there, but it's not infectious. It hasn't caused disease, you know, but it's being interpreted as because PCR told us it was. So it must be backed up with an antibody ELISA test or some sort of isolation identification. But even then, ELISA tests aren't without their false positives either. And as we know with TB, you get false positives with those too. And what they don't tell you is actually it's the cattle that are late in gestation, so they're getting close to actually calving. Those are the ones which are more likely to have a false positive. And the number of farmers where they're getting a positive on their farm, a lot of them are late in pregnancy, then it's quite highly probable that they are false positives. So as farmers, they should be asking what tests are going to be carried out and to make sure that you aren't exposing your animals to unreliable tests and unnecessary consequences, because it won't just affect you, it'll affect your neighbours as well, because there'll be exclusion zones put around too. But this needs legal advice as to where you stand legally. As far as I'm concerned, you've got an obligation for your animals not to expose them to a test, which could be a high incidence of a false positive. But obviously, you know, we need some legal advice, good legal advice on this as to where you stand on that. Because if you can stand there knowing that you're backed up by law, then you're in a much stronger situation to go, no, you can't come on my land because this test isn't reliable and we have evidence to show that it's not reliable, and therefore I'm not going to expose my animals to it. So I did some correspondence on this because I was pretty shocked that the PCR was being used. As it turns out, it's been used for a long time in veterinary medicine, but because I don't do conventional vet work, I wasn't using it and I wasn't aware that it was being used, but it is. So I asked the VMD, who are the Veterinary Medicines Directive, why they'd approved PCR testing for clinical diagnosis when they weren't considered suitable by Carrie Mullis. So they wrote back to me and said, there is no authorisation process in place for tests. Nobody official or independent has verified their efficacy or established the figures for false positives or false negatives in different scenarios. And that will alter depending on the disease prevalence, obviously. But basically, they said to me, well, why don't you ask the test manufacturer? Oh, well, they're going to tell me the honest answer, aren't they? So whilst it carried on, the VMD forwarded my email on to the APHA, DEFRA, and they said, they came back to me and said, well, their staff know what they're doing. And it's like, well, I don't care about the nuts and bolts of doing the test. I know you can do that. But the replies didn't reassure me at all that they understood the interpretation properly, such as the risk of false results. Because my email, as you can imagine, was basically saying, what's the risk of false positives here? They did not address that false positives issue at all. If they'd known what it was, you could bet the bottom dollar they would have come back and said something to me about it. And all this correspondence, if people want to read the emails, they're all on my website, and I'll give you that at the end. So in my opinion, the PCR is being used inappropriately and interpreted inappropriately too. We risk a situation if we're not careful, as occurred with foot and mouth in 2001, where thousands of healthy cattle and sheep were culled for no good reason. What is the end game? I've actually written a letter to the Veterinary Times, which I'm hoping will be published, basically saying, what is an achievable end game here? Because we're not addressing the reservoir in the wild birds, and I'm not advocating a mass cull of those either. But we've got to work out what is a reasonable and achievable end goal where we actually have still got farming and we haven't decimated our national flock without having the ability to actually control what we're trying to control. So moving on from PCR, we're going on to the modRNA jabs, which are inherently dangerous. So they are not vaccines. They are gene therapies, and that's included in their BioNTech SEC filing. They recognised it there, so it's not me saying it, it's actually in the company that developed these vaccines' own literature that they recognise that they are gene therapies. And I call them modRNA because it's not mRNA as we get in our cells. They've actually changed one of the bases in them so that it's not natural. So I call them modRNA just so that everybody knows what I'm talking about. As they redefine, when you've changed, they changed the definition of vaccine in order to be able to include these. And I say to people, if I redefine all small, furry, four-legged animals as dogs, it won't make my cat bark. Okay? So the lipid nanoparticles, which is what they used to deliver the modRNA into the body, they can cause inflammation themselves. They can cause emboli, so that's blood clots through clumping, and allergic reactions. And there's various different mechanisms they can do that. They do not stay at the injection site. Anybody who's given their cow antibiotics in the backside to treat mastitis knows where you put it in is not where it stays. The idea that any oil-based injection would stay at the site of injection was complete fallacy. And you know, we know that from all the other injections that we give, that they go body-wide. So especially they go to other organs which have got high fat levels, which are the gonads, so the ovaries and the testes, lymphatic tissue, and the brain and the nerves. And it's been shown now that the modRNA can enter the nucleus and can reverse into the genome, and actually the lipid nanoparticles make that 100 times more likely that those RNA, DNA will go into the cells, because that's their whole function, is to deliver what's in them into the cells and through membranes. And so the fact that we've got the cell has got a membrane and the nucleus has got its membrane. So the lipid nanoparticles will actually deliver far more, 100 times more into the cells than if it had been just injected without it, which is why they're there. But they've also shown this contaminant DNA that's in there also gets equally absorbed at that higher rate as well. So due to the pseudouridine, which is one of the bases that they've changed, the body doesn't always recognize it, and it doesn't stop the protein manufacture. They've now found that those who've had COVID jabs, 709 days in some people, they're still finding spike protein. So probably that isn't the original spike protein. It's probably because it's reversed into the genome, into the nucleus, and they're actually still producing spike protein on an ongoing basis. So they'll also, you're putting in strands of this modRNA, and of course the body will break it up, and that's what they've always said, that it will be broken up. But of course those shorter chain lengths actually have some biological activity themselves. They're called microRNA. Humans have about 5,000 microRNA, which are cell signaling, so they tell the cells and different instructions to the body what to do within the body. So the fact that you're putting something in which can be broken down into these microRNAs, and they've got no idea what they all do. They know probably about what 5 of them do. So if you've got 5,000 there and you don't know all their interactions and how they actually work together, they won't be working in isolation. You would need some sort of computer the size of a room to actually try and calculate how these will all work together. We've also got a prionogenic risk. And that's been shown to happen for the modRNA itself, and also potentially the proteins that they are instructing the body to produce. And there's been no work done to check whether any of the proteins which are instructed to be produced have any prionogenic activity. There's also a potential autoimmune risk. So if the proteins which get decided to be produced actually have any similarity to proteins which naturally occur in the body, antibodies to what they think is the pathogen will also then start to recognize self as a problem, and you'll get autoimmune problems happening. So the modRNA in animals, because this is my concern, is obviously where they're going to, obviously I'm concerned about what happens to people, but I'm also concerned what's going to happen with our animals. So there is currently one modRNA product for pigs in the USA called Security. And it's actually been ongoing since 2018. How they got it past the vaccination definition before the definition being changed, I don't know, but it just shows the level of corruption in the authorization processes. So Security is a platform, as a product platform, not a specific end product. So if I tell you what happens, so you've got an outbreak of, argument's sake, respiratory disease in pigs. The vet comes along, they swab the pigs, they send the swab back to the lab sequence it, they choose a gene of interest, and they make a modRNA vaccine for that, and then they send it back to the farmer to inject. Guess how long it takes from swab to coming back to the farm as a vaccine? 8 to 12 weeks. But you can't check anything, especially things like prionogenic activity, in 8 to 12 weeks.
Can you say what prionogenic means, please?
Yeah, I'll come on to prions in a minute. So prions are basically what cause BSE. So Creutzfeldt-Jakob disease, they are misfolded proteins which are infectious themselves, but what they're like seeding a crystal layer, once they go in to and start this misfolding process, it causes all the proteins around them to do it. So it builds up a plaque, if you like, and that's what then causes the neurodegeneration that causes a spongiform encephalopathy. So you get the brain and nervous tissue looks a bit like a Swiss cheese with all the holes in it, and that's why it's called that. And it's caused by these prions. So the declared intention is to move animal vaccination to modRNA jabs. It hasn't happened in the UK yet. That's one positive. They have an inherent risk and they aren't being checked, like the prionogenic activity. If a chosen protein or the selected modRNA itself is prionogenic, then it will initiate the protein folding and the prion formation. Prion shedding can affect the microbiome and then the environment. If an infected animal is slaughtered before symptoms manifest, the prions will be in the food chain and could potentially make meat for food infected for humans and animal carnivore products themselves. So dogs and cats, obviously carnivores, so they should have meat. And so the RNA and prion hazards. So the modRNA itself, as I said, can be prionogenic. The chosen protein can be prionogenic. So you get frame, and this happens because of frame shifting errors in terms of how the protein is made from reading the RNA that's gone in. And you can also get different areas on the protein have the potential to be prionogenic in that they don't look at. So looking at it from one aspect of a single immune response, but they forget to check or look at the other parts of the protein to see whether they could have a different action elsewhere in the body too. So these proteins may be shed in the milk, sweat, urine, feces and saliva to the environment, and the prions may also shed as well. Prions in slurry used as fertilizers can go to the soil and soil fauna, so like worms and beetles and all the rest of it potentially. And the latest research shows that plant roots can uptake prions from the soil and accumulate it in leaves, which obviously if you're eating a crop, you're going to be eating it in your plants as well. So those of you who are perhaps vegetarian or vegan think that you've got nothing to worry about, got news for you, unfortunately, potentially you could have. So the prions in the soil will leach down into the water system. They're not neutralized by normal water processing. They can potentially spread, maybe same as microplastics can in rain. Who knows? I don't think the work has been done to show that. There's been a lot of question marks over how these, especially chronic wasting disease in the States is a big problem at the moment. But it started around some of the government laboratories. I'm quite sure how that happened or why. But so this is just a diagram of what can potentially happen. So you've got the modRNA, and it obviously gets the body to produce proteins under instruction. And then you've got this PRPC, which is the normal form of protein in the body, and the action of either of them can make it go to the scrapie form. Scrapie is the sheep version of mad cow disease, the spongiform encephalopathy. And of course that could then potentially go to the topsoil, could potentially go to the water, can go to the plant roots, can go to fruit and veg, or go to the grass and grazing, and then can go to chronic wasting disease in deer, or obviously BSE in cattle or whatever else is eating. So potential consumer dangers. So consumption of prion-infected meat, because it doesn't just go into the nervous tissues, what we thought 20 odd years ago when BSE first started, it will actually go into the meat as well. So be wary if you're shooting wild deer in the States, or obviously if it's going to farms, if these jabs were to be used in the farms, we've got the potential that it could go into our human food chain. Via the crops as well, and potentially into the water. And potentially dairy produce as well. The other thing is, which is important because I'll talk about, they're looking at changing the rendering process in proteins, which I'll talk about shortly. It's important to note that the consumption of prions that pass through another species can have accelerated misfolding activity. So, for example, if a BSE cattle prion went into the soil, went into the plants, the deer ate it, and then someone ate the deer meat, obviously that because it's a bovine cattle prion, it won't cause disease in the deer. But if someone were to eat the deer meat which had the BSE prion in it, the activity or the path of what happens in the person who ate it would be accelerated compared to if they'd had the beef itself. So this is an important point to note. So there's a risk of new and previously unknown prions, e.g. super prions, so they wouldn't have any species specificity. It would be across the species barrier with no problems at all. Just as an example of how dangerous they are, previously unconsidered risks from surgical instruments. So they had, with Creutzfeldt-Jakob disease, which is the human version, they actually had some patients where they did some brain surgery on them, and obviously all the surgical instruments get sterilized between operations, and the person who had the next operation following the previous one, they actually got Creutzfeldt-Jakob disease from the sterilized surgical instruments. So that's an example of how indestructible they are potentially. So we've got to have considerations for blood transfusions, organ donation, and exosome shedding as well. If it becomes rife, see, we can take a bit of a breather now. We're not in this situation yet. I'm warning where it might go to, so don't get overly concerned yet, but this is why we need to keep an idea and keep a close view on what is actually potentially going to happen. And also you can get interference with P53, which is the body's own natural cancer suppression. So it's our protective cancer levels that we have, all of us have got it in it. So if you interfere with that, then obviously you can get things called turbo cancers developing. So what's the difference now compared with what happened with BSE back in the 1990s? So if they start introducing these modRNA vaccines for a whole herd inoculation, massively going to increase the chances of producing prions. And it's not really going to be a question of if, it's probably going to be a question of when, simply because the numbers involved make it far more likely. Even if it was a 0.0001%, by the time you've injected 100 million animals, that is actually quite a significant number. It becomes a game of numbers. As I said, they're highly indestructible, but they will tend to then accumulate and concentrate in the environment and also in the animals potentially as well. They're impossible to filter or neutralize them. So as I say, the sheer numbers of animals involved will make it greater the risk. And also bear in mind, with the WHO One Health policy, they can potentially mandate action in animals as well as obviously what they can potentially mandate in or try to mandate in people. So think of the pandemic treaty that they're trying to push through. There is the potential within that it could even affect your animals if they say, right, you've got a zoonosis, which is a disease which can be passed from people or from animals to people. If you've got a zoonosis, therefore we're going to mandate these mRNA jabs, and the WHO could potentially do that if it gets passed and if it's allowed. Hopefully it won't. And given people's reluctance now to take the modRNA jabs themselves, and given the existing government contracts, animal use could be seen as the contractual solution to this, and could be pushed under the excuse of countering antimicrobial resistance as well. So in the same way that they changed the definition of a vaccine to allow the modRNA, they could say, well, this one, because we're using it against a bacteria, we're going to call it an antibiotic. So just be careful. It's not happened yet, but it is a potential risk down the line. So obviously, a lot of us have got animals and pets as well as our farm animals. So there are already modRNA jabs being used in North America primarily already. So they've got a rabies one in Canada, canine influenza in USA, Mexico, Chile and Philippines, which came in June 2024, and there's a cat FELV, feline leukemia virus, in the USA. It started in February, so it just happened in 2025. They're not in the UK yet, but these are made by MSD now, and they're now calling them RNA particle vaccines. So they're changing the name to try and separate out from all the bad press, obviously, the mRNA jabs have had up to date. And they're under the brand line name Nobivac NXT vaccines. That's how they've been branded in the States. Dare say if they came or tried to come elsewhere, they would use a similar sort of brand name. Given that shedding has been proven to occur with the COVID jabs, I wonder if anybody has looked into these for the animal side of it as well. I suspect not. There are also self-amplifying modRNA, where the body is directed to produce the mRNA itself in an uncontrolled manner, which will then produce the protein in an uncontrolled manner. So you've got two levels of uncontrolled amplification potentially going on. Again, none of these will have been checked to see whether they've got prionogenic activity or any autoimmune issues as well. Obviously, when our pets, we hope they're going to live long, healthy lives, so the chances of some of these diseases actually manifesting will be much higher in our pets than we'll potentially see in animals which are being grown over a much shorter time period for meat. They won't necessarily have time to show and manifest these problems. But we would see them in our dogs and cats, unfortunately. What could possibly go wrong? So the consequences. So there's no safe level. What's happened to number 2? Prions and amyloid, which is, again, another protein misfolding. You get cardiac amyloidosis and renal amyloidosis. So it's a protein misfolding that happens in other parts of the body. The enzymes can potentially break them down, but the breakdown products themselves can have potentially prionogenic activity too. So breaking them down isn't necessarily the best idea, because all you do is end up seeding it around the body. So potential treatments need to be properly assessed. Don't dive into something just because someone says, this breaks down, prions on the internet. It's like it may do, but just be careful what you do, what the consequences of that are doing. It just needs to be properly assessed. So their resistance to heat, we've already done that one, and all treatments will work for everybody. And the delay in disease onset means that some of these treatments may start too late because it's already reached a certain level of folding before the symptoms manifest. And if you think this is all fanciful and that there's nothing to back it up behind it, I'm sorry, but I've got some bad news for you. They've done a prediction. They've got some actuaries or whatever they do within the pharmaceutical industry to look to see what the future of prion treatment is going to be in the future. And as you can see, from 2023 going up to 2030, it's massively increasing. So they've somewhere have got an idea that prions are going to become a problem in the future, because huge investments will be put into developing drugs to treat these on the background of this. So this isn't my speculation. This is something which the industry themselves are already looking at. Otherwise, they wouldn't come up with this graph. The same with Sandy, everything I put up, I try and make sure that I can back it up with something that is either from their own literature to prove that what we're saying isn't speculation. It's actually got some basis and fact behind it. So this might be a bit of a shock for some people, but the processed animal protein inclusion rules are looking at being changed. So DEFRA has a consultation which is ongoing now, looking on relaxing the PAP rules, and that, funny enough, they've chosen April Fool's Day for the consultation to end, so I'm not quite sure who's being played for the fools here. So this would basically allow poultry pap into pig feed and pig pap into poultry feed, on the basis that both species rarely show transmissible spongiform encephalopathies. As I've already said, when you've got a prion passing through a species that it doesn't supposed to show in, but you're also chosen a species where it tends not to show, you've got even less warning that something is coming through that process. So what they think is actually a safety net actually may be a hidden opposite. They also intend to allow insect pap into pigs and poultry feed, and ruminant collagen and gelatin in non-ruminant animals as well. And the reason for justifying this is because it brings us into line with the EU, but I thought we'd left that for a reason. That is not a good enough reason to change our regulations. It's got, let's base this on science. If we found that what we did with the protein rendering rules actually worked, then if it ain't broke, don't fix it. It's just common sense. So it seems to me to be the perfect way of potentially developing a super prion, that where we wouldn't necessarily see it until it's already been transmitted to people. Again, what could possibly go wrong? So here we go on to Bovaer. So I think everybody's probably heard of Bovaer. They've seen the Arla tweets and all the rest of it. So currently Bovaer is licensed in the UK as a feed additive for lactating cows. Licensed for beef in other countries. And the active ingredient is called 3-nitrooxypropanol. Propanol. It's also got silicon dioxide and ethylene glycol in it. And it inhibits an enzyme, methyl coenzyme M reductase in the rumen, which is the big stomach where all the grass ferments normally, for those who are not farmers. But no one's done any studies, as far as I can see, to see what effect that might have on the microbiome, which, as we know, is hugely important for immunological health and also mental health as well. And I don't believe that animals have any less implications for their mental health than the humans do, so apart from the immune ones as well. And there's also other effects that we're discovering with the microbiome all the time. So we just really have no idea when the microbiome is 90% of the organism. Once you start introducing changes and imbalances in that, we really don't know what the long-term effects of doing that are. According to its data sheet, you need full PPE for handling it because it's a skin and eye irritant, and they're not the same, and it's harmful if inhaled. So it's been passed by the Food Safety Authority and FSS in Scotland, Food Safety Scotland. So there's been no VMD assessment for the animal health and welfare. And I've had that in an email from the Chief Veterinary Officer, because I wrote to the VMD and I questioned why they hadn't authorised it as a pharmaceutical like the FDA had. So the trials are relatively short. There's only 15 weeks is the longest trial period where cattle were fed Bovaer, which obviously consider that relative to a 305-day lactation and then multiple lactations in a row. We haven't got any long-term data. So I don't care how scientific someone is sitting on their committee, if you haven't got the data, you can't analyse it. And that's the bottom line. What they did find, however, that some of the ovaries became smaller. There was unsure about the genotoxicity, which is obviously the effect on the DNA. And high doses caused cancer in rats, but we don't know, obviously they're not going to feed that high dose, but we don't know what the cumulative effect is because they haven't done the studies for long enough to see whether there is some sort of accumulation of that. There's been some unconfirmed reports from Denmark where it is mandated from farmers who have been forced to feed Bovaer but who daren't speak out, that it causes diarrhea, fever, induced food intake, and also therefore milk drop, and increases mastitis, probably again low energy, immune system depressed and all the rest of it. So the idea that it has been assessed and found to be healthy is obviously completely wrong. The typical UK diet, a lot of this work was obviously done in the States where there are a lot of grain lot cattle over there, much more so than we tend to have over here. And the diet that was typically fed in the UK has got much higher level of a certain type of fibre, which means that the Bovaer has an even less of an action anyway. So, what are we trying to achieve? So moving on to, it takes us into climate, starting off with methane. So methane is only present at 1,911 parts per billion. So it's 0.0001911% of the atmosphere. Some insects also produce methane. E.g. termites alone produce 20 to 30 tons per annum, which is compared to the ruminant 120 odd tons. So you've got one species producing a quarter of the methane that ruminants supposedly produce, and that's only an estimate as well. So, and there'll be other reasons why it's being produced. So the rumen, obviously we've got this big fermentation vat where all these plants are fermenting to create the energy that feeds the cows. And it's never been a problem. And it's going to produce methane, which is then going to go into CO2 and water, which are supposedly greenhouse gases as well, but even worse than methane. So all we're doing, a bit like Sandy was saying, how they moved coal and steel and production from the UK over into China and India. All we're trying to do with Bovaer is move it from inside the cow to outside the cow, and then saying how great we are because we've done that. And it's like forgetting what damage it does to the cow in the process, or potentially the milk. They say it's safe, but again, how long have they done the studies to prove that on a cumulative basis? So methane was never a problem when huge herds of ruminants roamed the planet. It's got a 10-year carbon cycle anyway. So the hydrogen that does produce will become water vapour, which are supposedly worse greenhouse gases if they exist outside the rumen. So the methane is broken down as part of this 10-year cycle by methanotropic bacteria in soil, trees and water, and it's sequestered in the ice. So it's released by the warming, the same as a lot of CO2 is sequestered in the sea. It's why when you have a glass of champagne, the bubbles get more as it warms up in your hand. The bubbles keep coming because it's the gas coming out of what was sequestered in the drink. So in the same way, when you've got gas sequestered in ice, in something cold, as it warms, then it's released. And that's the order that it happens, not what they tell us. If we look at the cattle population graphs, they don't match the methane graphs either. There we go. Perfect match. Can't you see cattle are obviously to blame for this change in methane? And that's exactly over the same number of years. So Earth temperature is primarily determined by our position relative to the sun. Who'd have thought it? The sun's solar activity, we get cycles of solar activity, and adiabatic heating, which is the pressure from all the atmosphere on the Earth. And that's what creates our temperature. The sun itself has cycles of activity, such as the 11-year sunspot cycle, that are not included in climate models. The Earth has other cycles, known as Milankovitch cycles. They're not included either. Our geomagnetic field, which protects us from excess solar radiation, that also will have effect as to what radiation gets through, as to what heating effect you get on the planet too. Consequently, comparing annual temperatures is not comparing like with like. So the original hockey stick graph that was used to justify this climate alarmism is a snapshot in time. They've taken a very short period of time to justify what they think they want to do. And it's left many questions unanswered. And the original authors refused to share the original data. That's how much scrutiny they're prepared to be exposed to. So you've got to say, well, if you're not prepared, if you're that sure of your theory and the data was that conclusive to come up with that conclusion, you would be happy to share your data, but they aren't. Why not? These are the Milankovitch cycles. So you've got one is eccentricity. Basically the circuit that we make around the sun, that will change. It will expand and contract in certain cycles as well. We've got the tilt of the planet, that also has its changes and it goes in its own little cycle. And there's all these other Milankovitch cycles which change how we are orientated to the sun, which will change our climate. So let's going on to climate myths themselves. So CO2 is only currently at 420 parts per million, which is 0.042%. Well below the 6,000 parts per million when Earth was at its greenest, and much closer to the 150 parts per million below which life cannot survive. And as I've seen recently, there's now question marks as to whether the 280 figure that they had just before the Industrial Revolution is even accurate itself. How convenient. There is no planetary greenhouse effect. Convection determines the Earth's temperature regulation. All climate modelling is therefore based on relief. There is no greenhouse effect. There's no radiation effect. Because if you think about it, the heat hits the ground, then it bounces back. And apparently what the greenhouse gases do is they bounce that heat back. But actually, if heat hits a molecule and it radiates out, do you think it just goes back to the ground, or do you think maybe 50% of it carries on out in another direction? So we only get 50% bounce back, and then that bounces back up again, and another 50% bounces back, and another 50% goes out. So over time, all the heat that supposedly the greenhouse gases are keeping in is actually being radiated out as well. But that is all happening at the speed of light. So it's, you know, you might get a delay in temperature or heat radiation from the Earth by a few minutes, but that's it. There is no heat. But yet, if you think about it, on a summer day, you've got all the heat, and if the cloud is there, then it stays like a warm, muggy night, doesn't it? But if the sky is empty and there's no cloud, it gets cooler. So it's not the gases which are changing the heat, what's heating the Earth, because otherwise it'd be consistent every day, every night, whether it was winter or sun. What's actually changing it is the cloud cover, it's the convection, and this is makes all these ideas of net zero, and Sandy didn't mention it, but there was actually an absolute zero behind that as well. So there was actually a government investigation or paper where they're saying they actually want to get down to absolute zero. And when they say that they're supporting farming, one of the slides that Sandy had, they want to get our meat consumption down to 0 kilos per person by 2030, but they think that might be a bit too much. So they're going to go for 16 kilos per person per year, which is and 0 dairy. So when governments say they are supporting farming, how can they say they're supporting farming when their ultimate aim, whether they achieve it by 2030 or 2040, is immaterial? If their ultimate aim is to get meat consumption down to 0 and dairy consumption down to 0 because they want all these other food patches and jellyfish that Sandy was mentioning, you know, it's totally disingenuous to say that the government is supporting farming. They aren't. So, as I've already said, temperature rises precedes CO2 because as the ice melts and the seas get warmer, supposedly, then they release their gases, and that's what creates the increase in CO2. It's based on poor modelling. The real data does not support the theory, and there's actually been evidence where they've been going around and doctoring the early data to try and make it look what they want to make it look like. Only 3% of the CO2 emissions are man-made. You show me any system where 3% of what is produced governs the whole system more than the 97%. I don't think it exists. We need the increased CO2 for plant growth. In those greenhouses all over where they're increasing the CO2 in those buildings to increase growth and yields. And we need the cattle for soil. We need it for regenerative farming. Alan Savory in Africa has been rewilding and greening areas of Africa by having nomadic herds which leave their dung and eat the foliage there. And that actually has caused all this regreening of the planet. So we know that we need it, and obviously a lot of the land that's being farmed is suitable for growing grass, but it may not necessarily be suitable for growing other more intensive crops. And we know that monoculture actually decreases all the nutrient value of the food over time because we're depleting the soil so much. So we need to get back to regenerative farming. So this is actually a graph over millions of years of CO2 and global surface temperature. As you can see, we're sort of on that far edge of the graph, so you can see where it's been before. So the idea we're getting to a point where the planet is actually under danger of from where we're heading is complete myths. And of course, this is the rubbish that they're teaching the kids, so that kids are genuinely fearful of where it's going. And this shows how, as Sandy was saying, this is all infiltrated into the education system as much as what we're seeing in the real world too. So going on to I'll rush through the solar and farm myths. So it's not cheaper, bills are still rising. We need a parallel energy source for when there's no wind and when there's no sun. They cannot replace fossil fuels. We need them for mining, infrastructure and construction and maintenance. It's funny how mining coal is bad, mining lithium good. These products can't be recycled, so they have to go into landfill, they're potentially toxic. And even if there's damage, you get these huge hailstones of solar farms, you get all the toxic leakage into the soil, which is just convenient because then if the farmers don't want to put the money back into another solar farm, they can only be used fit for growing, for building on, because they're no longer suitable to go back into farming, but we need our food. So they harm birds, whales, soil organisms, so they're not exactly green. And there are better energy technologies on the way. So we've got the thorium. China has just found a huge thorium source. They reckon that they could provide electricity for the Earth for the next 60,000 years just on this one source that they've discovered alone. And they've got a thorium reactor coming online in the Gobi Desert in the next couple of years. So it's not that the technology to use it isn't available either. We've also got hydrogen. Malcolm Bendel's thunderstorm generator, you can look these up on YouTube, and also Maxwell Chikumbutso's microsonic energy device, and he's planning on driving from Harare to somewhere else, a very long Cairo, I think, some very long place far away, without having to refuel, because he's basically taking the energy from the air. So, and when he gets home, he says you plug the house into the car so that you then power the house, not what we're doing it the other way around. As Sandy said, the energy we need for all these data centers, AI, Google and Amazon said they need 4 nuclear reactors just to power their future AI requirements. So you can imagine where the priority, if Keir Starmer says he wants to make UK the centre of a leader in AI, well, you can guess where we're going to be on the priority for electricity when it comes down to it, assuming we can even produce it, Ed Balls. And what a balls up that would be. So we really need to sort of stand up and recognise this for what it is and talk about it. That's what needs to happen. I did a talk to some farmers in Exmoor, and one of the farmers came up to the end and he said, "Great that you did the talk." He said, "You've been saying what we've been thinking individually, but we were too scared to mention because we felt isolated." And so what I would say is talk to people. The biggest way this message gets out is by face-to-face talking. And there's actually an app you can get on your phone that says, it's called Inconvenient Facts. And you can get it on your app and you can go, look, this is the myth that they're basing this all on climate. And this is what we need to overturn, because it's going to ruin us as a country economically and food production. And if you farmers stop producing food, you're the ones that are going to go hungry the same as we are. One last thing, all the correspondence that I've had with the Veterinary Medicines Directive and also with the over Bovaer and the PCR test and the modRNA is all on my website. I did an open letter in 2022, I think, with Kevin McCann. Some people might have found him online. He's a systems neurologist expert, and he helped me write my concerns about prion production from modRNA use in animals. So all the correspondence is there. So if you want to check my interpretation, you can do that too, and all the evidence to back up what I've said. I've got leaflets at the back too. Yes, thank you.
The massive dangers of scientocracy and where it could lead us if too much heed is paid to it. I think that was a phenomenal canter through a huge volume of things that we're very we are now able to use a huge amount of information that was imparted and strengthen our position because of it. But I think also to highlight, particularly, think of what the Veterinary Medicines Directorate is incapable of doing, which is following its own processes or indeed having any form of rigid background to them, and how that can easily run out of control. And then you see at the other end John Magufuli, who was referred to as sending off fruit to be PCR tested, who the then president of Tanzania, interestingly, politically refused to implement any of the measures that were being put in any other parts of the world, and he died in March 2021. The current incumbent, of course, went along with all the various measures that were being implemented everywhere else, so that's another point worth noting. And again, I think just thinking of how modelling works, not just with climate, but in terms of the predictions for disease, and lo and behold, the people who are able to predict that are the people who can tell you that you have a disease in the first place. So the testing mechanism as a weapon is something that I think Roger has really brought out, especially with PCR.
Yeah, and Niall Ferguson, of course, is the common name with all of them.
Absolutely, going back to 2001. So a huge amount more to look into. I think his closing note that this is all about face-to-face communications, and that's why we're here. And this is significant in its own right, and it will be beyond today. And not to make too much of a thing of it, but I think it is worth noting, just down here to my left is Sir Geoffrey Clifton Brown and his wife, Lady Clifton Brown. He has been a serving MP since 1992, and he is engaging in this debate by simply being here. So that is worth noting. This stuff is being done.